All right, so we're going over the chapter 1 PowerPoints, and I'm going to focus on the important aspects of it. And I mentioned the first PowerPoint is very general. It introduces you to concepts of microbiology, including the history of microbiology, which is a very young science, barely over 100 years old, versus thousands for other sciences, like math, for example, because that's not a science. But, well, even anatomy and physiology, you could argue, is almost 1,000 years old, technically speaking. So it is a very young science, microbiology. So focus on the important players like Van Leeuwenhoek and Tyndall and Koch, and obviously Pasteur. Know terms like what is a germ, know the difference between the theories of spontaneous generation versus biogenesis. And the reason why we spend some time on Louis Pasteur and his experiment that debunked spontaneous generation is because it is an important exercise for microbiology. And so be familiar with the what's called the gooseneck flask experiment that Pasteur conducted to show that spontaneous generation did not hold water, that biogenesis was the correct theory. And so the reason why this is an important microbiology experiment is because it, first of all, it brought to the forefront the fact that microorganisms are everywhere. So Pasteur was operating under the knowledge that there were microorganisms in the air. That was the reason why, if you had a flask with clean, clear broth that had nothing growing in it, but you left it open to the air, that broth would become contaminated. So in order to prove that you needed microorganisms to make microorganisms, he had to prevent these cells from reaching the broth, and he did that by creating these flasks that were actually I do have access. Yeah, these flasks that had these curved necks, and the dust, air as it came in, the dust was trapped in the neck of the flasks, in the curves, and so what went into the flask was essentially filtered air, devoid of microbial cells. So, by the way, the flask had been the broth had been heated previously to make sure that nothing was in the broth to begin with. So, first of all, what was he showing? He was showing that we could kill cells with heat. Live cells could be destroyed with heat. So we could manipulate their presence or their absence, for that matter. So we could let them grow with a little bit of heat, warm temperatures. We could kill them if we applied excessive heat. We could filter. We could prevent their entrance. By physical means, in this case, by creating these curves in the flask. And so the experiment showed that if you prevented these cells from entering, then nothing would grow. So this is a manipulation of microbial growth. That's what he's doing. That's why this experiment is so important, because if you want to study something, you have to be able to manipulate it. You have to be able to bring it into the lab and manipulate their growth. And that's what he was doing with this experiment. So he opened the door for that. He opened the door to microbiology experiments, to bring in microbes into the lab. So be familiar with the gooseneck glass experiment. And this is a rundown of what is so important about this experiment. You know, we can control their growth with heat in both ways. We can make the heat, have warm temperatures, and that encourage the growth. We can have extremely high temperatures that will kill them. Also we can identify their growth in substances, because those substances, the liquid substances, becomes cloudy. And so as you're going through your normal life while you're taking this class, pay attention to where you find microorganisms. And oftentimes in the kitchen, I mean, I don't know about you guys, but it happens to me, sometimes food goes bad. And when you look at the food and you decide whether it's bad or good, one of the things you do is you smell it. And you know, microorganisms are producing chemical substances that are changing the environment and creating, chemicals, smells, that breakdown of substances. Also, sometimes when food goes bad, it's obvious because you see the mold, you see the fuzzy things growing, and that's mold. Okay, but other times food goes bad, and instead of seeing the fuzzy mold, what you see is slimy, slippery stuff growing in the food, and that's bacteria. Okay, so I don't know, maybe gross to some of you, but pay attention to that. To the difference between bacteria growing versus other kinds of microorganisms like mold, for example. And also aseptic techniques, which are the techniques that allows microbiologists to work with microorganisms in the lab, because aseptic techniques prevents the growth of unwanted microorganisms. So when you start from the premise that they're everywhere, but you have to control what you have, when you have it, whether you want them or not, then you need to be able to prevent the growth of the unwanted ones and encourage the growth of those that you do want. So that's what aseptic techniques allows us to do. And aseptic techniques is what we use in a clinical setting to prevent contamination of clinical devices, to prevent a patient from getting sick, for example. We follow aseptic techniques all the time in microbiology. When you cook, you're following aseptic techniques. You're trying not to contaminate, cross-contaminate foods like meats and vegetables, or cooked foods versus non-cooked foods. So you are aware of the presence of microorganisms, or you should be, and are trying to prevent the growth of unwanted organisms in places where you don't want them. So aseptic techniques allow us to isolate microorganisms out of their natural environment and bring them into the lab, grow them in the lab, culture them, identify them, and then study them. And this is the classical approach to study microorganisms, which is not perfect by any means. But that's what we have. Aseptic techniques know what it is, know the definition. And there are several definitions, but the principles are the same. By the way, feel free to interrupt with questions. We talked about Kendall before. As one of the people that you need to know what their accomplishment was. And he is the one that you could say discovered the presence of endospores, bacterial endospores. And basically what happened was when Pasteur conducted his experiment, for an experiment to be valid, it has to be reproducible. So other scientists were trying to reproduce Pasteur's experiment. And scientists were not as precise back then as to how they did things. So Tyndall was trying to do the same thing as Pasteur, except he was not using the same medium as Pasteur. Pasteur was using beef broth that had been boiled. And even to make beef broth, you have to boil the meat and all that. What Tyndall was using was another way to gather microorganisms, which is to make something called a hay infusion. And turns out if you take hay, and for that matter, it could be grass, just dead grass, and you put it in water, give it a few days, and the water will become, even become cloudy, but it will have tons of microorganisms growing in it. So that was another way in which scientists were creating these substances with microorganisms, was to make hay infusions. And that's how Tyndall was doing it. He was creating a hay infusion. He was using the hay infusion, then taking the liquid from the hay infusion, boiling it, and then following on Pasteur's protocols, except that his protocol, it wasn't working for him. The substance, the broth, did not remain microbial free. It would grow microorganisms regardless of the curves of the flask. What he finally figured out is that boiling was not killing all the forms of microbial life in the hay infusion. He figured out there were forms of microbial life that were heat resistant. He called those forms endospores. And this is what happens. Turns out there are bacteria who are able to create dormant cells called endospores. So the live metabolically active cell is heat sensitive, we call it a vegetative cell, to distinguish it from the form of a cell that some microorganisms may form, some of them may form, under environmental stress, which is an endospore. So it turns out some bacteria, not all, when you put them under an environmental stressful situation like heat, like high heat, dryness, radiation, these cells have the ability to go into a dormant state. They dry up, they stabilize their DNA, and they become a non-metabolic cell. It is essentially a dormant cell that will remain so for as long as the environment remains unsuited for life. It could be years, it could be hundreds of years. There's reports of these endospores isolated from honey vessels that were hundreds of years old, and they were able to make them come back to life. So they are extremely resistant. And again, I'm going to emphasize some bacteria can do these, not all. Anyway, so the trick is that many of the bacteria that can make endospores live in dirt. And Tyndall was using essentially hay, which had dirt in it, to make his broth. That was his problem right there. So he was collecting bacteria that were endospore formers. And they were making endospores, and endospores are not killed by heat, are not destroyed by heat. So he would boil the solution. The solution will come back to normal temperature, warm room temperatures. The endospores will now come back to life as vegetative cells, and they begin to reproduce. So endospores are important because in order to make a solution sterile, you need to make sure you use a means that can destroy endospores. So endospores are tough, but they are not completely indestructible. We could destroy them if we have high heat, pressure that will destroy an endospore. So Tyndall discovered the presence of these other forms of bacterial life. And when we do the staining lab, we'll do an endospore stain, so you can see the vegetative cells, the live cells, versus the endospores. They're very tough structures that do not stain easily, so you need special ways to stain them. And this is just to emphasize that we could determine bacterial growth by looking at the cloudiness of a test tube. So test tube B looks clear. That doesn't mean there aren't any organisms there. That only means there aren't enough to make the tube cloudy. By the same token, the cloudier the tube, the more bacteria you have. So that's one of the things we miss by not having face-to-face labs, is that you don't see this bacterial growth in test tubes. Okay, next PowerPoints just go over how important microorganisms are, and I'll let you read that. It's not necessarily too hard to understand. Do know the terms like biodegradation, biotechnology, genetic engineering. This is an important term. I may not seem so to you, but it is. The concept of a pathogenic organism. And I'll come back to this in just a second. Yeah, this is just going over, this is going over, medically important microorganisms. Keep in mind, and this is also, again, historically very important, is the fact that modern sanitation alone, that was probably one of the greatest achievements. And we don't see it as such. We completely gloss over it, and we don't realize that that is the reason why we don't have the widespread diseases that we used to have, is due to modern sanitation, meaning running water, the sewage being treated properly, having drinking water, healthy drinking water, clean streets, clean homes. That alone is the biggest achievement that has prevented, that kept a population healthy. Then you can have the secondary ones, things like vaccination, antimicrobials. And the problem with the secondary ones is that they're not perfect. Whereas modern sanitation, I mean, you can even argue that one may not be completely perfect, but it's still, there are two sides to antimicrobial, the use of antimicrobials, because they have brought about problems like resistance to antimicrobials. Okay, so will dates be important? No, they will not be important. You do, I mean, this is, you need to have a sense that there was a pandemic at the turn of the first century. You probably have heard about it by now, because this is 100 years later that we'll have in a second pandemic. But no, you're not going to be asked what years they were or anything like that. No. Know what the germ theory of disease is, which is exactly what the term implies, is the theory that says germs cause disease, which to us would say, well, yeah, but you have to prove that. You cannot just blankly say, oh, germs cause disease. You have to prove that that is correct. And so there were experiments, there were people that worked on proving that, so that as to convince everybody that indeed germs cause disease. Let's see. So we talked about a little bit of antibiotics, vaccines, some of the epidemics, and things like plague were others were conquered through sanitation. Smallpox was conquered through vaccination, and do have an idea of when it was completely conquered. This has been the only disease that has been conquered through vaccination. The next one is polio, but still we have cases of polio, very few, maybe one or two reported every year. So it's not completely eradicated, whereas smallpox has been eradicated. So again, I'll just let you read about the importance of all of these achievements. Do know the difference between emerging diseases and newly emerging diseases. Emerging diseases are the new ones. And here, let me explain these. When we say new, we do not mean yesterday. We mean 200 years is new. Okay, keep in mind the old diseases, the ones that have been with us for thousands of years. Tuberculosis, syphilis, even smallpox, we know we eradicated, but before 1977, smallpox has been with us for hundreds and hundreds of years. Pneumonia, all these are old diseases. New diseases, maybe as new as 100 years ago or 70 years ago. Lyme disease was discovered in the 70s. Legionnaire's disease, I believe that one was late 70s also, or early 70s. AIDS, HIV in the 80s, obviously Zika, COVID, all these are fairly new, just a few years ago. But I just want to point out that a disease that came about in 1970 is a new disease. Okay, most of you were not alive in 1970 and you think of it as an old disease. It's not. Okay, it's a new disease. So do know the causes of emergent diseases, like changing lifestyles and genetic changes of the organisms themselves. And notice this is a map of showing you some of the new diseases. And again, some of them you may say these are not new at all, but when the time frame is hundreds of years, then yeah, they are new. So I'm just trying to see one of the like hantavirus, I don't know what Lyme disease is. Oh, Legionnaires, 1976. They don't have Lyme disease in here. It should have been, but okay. Now, new diseases versus re-emerging diseases. Do know the causes for re-emerging diseases, what brings them about. There's about 5 of those causes, so do know that. And also be able to come up with the examples of re-emerging diseases. And those will be things like measles, for example. And that's a pertussis, whooping cough, maybe another one. And then just new challenges in the future, okay. All right, this is another thing, another PowerPoint that has important concepts. The concept of normal flora or normal microbiota, they're both the same thing, different terms, but they mean the same thing. These are organisms that live in our bodies, and they are healthy. We need to have them there. They're incredibly important, and the more we know about them, the more important they become. they are probably the new frontier of medicine, is to understand the role of normal microbiota and normal flora in human health, which is huge. Linked to things that you would never think they would be linked to, like Alzheimer's, for example. So, however, having said that, normal flora are also opportunistic pathogens, meaning that in the wrong place at the wrong time, or in the right place at the wrong numbers, they could cause disease. So, given the opportunity, they could cause disease. So, two concepts here: the one of normal flora or normal microbiota, what are they? And the concept of opportunistic pathogens. So, opportunistic pathogens versus true pathogens. One of the first huge difference between them is that an opportunistic pathogen is common versus a pathogenic organism is rare. So, opportunistic pathogens, E. coli is an opportunistic pathogen. It lives in our colon, safe and good to have there. However, if it travels to the urethra, it could cause a urinary tract infection. All right, that's an opportunistic pathogen. It's common and causes opportunistic types of infections. A true pathogenic organism would be something like the Mycobacterium tuberculosis, which causes tuberculosis. It is rare in the environment. You can say right now, you probably have E. coli somewhere in your house. I can assure you that 100% on some surface, on obviously in some part of your body, but it is common. Mycobacterium tuberculosis is extremely rare. Okay, so it goes beyond whether they cause disease or not, is the fact that one is common, the other one is rare. So a pathogenic one is a rare organism that causes disease in any host, and that could be any host, that could be debated, but okay, there's a definition, that's fine. Typically, an opportunistic pathogen is common and typically causes disease in weak hosts. Okay, so that's an important concept that is going to be with us the entire semester. So do know the definition of these terms. A normal flora may be opportunistic. However, there may be strains of normal flora that could be considered pathogenic. So E. coli may be considered an opportunistic pathogen. However, E. coli 015787 is a strain of E. coli that is considered pathogenic. So that's another concept we're going to revisit. So that's it for the first PowerPoint. Let me go ahead and open the second PowerPoint. And I would say the second PowerPoint is probably more important in that it's going to be with us the entire semester. And what it does is it gives you a survey of microorganisms and their characteristics. So it starts by introducing the domains, the living domains in this planet, in the living world. And there's 3 of those. And all 3 would have microorganisms. Domain is the big classification. Domains are subclassified into kingdoms, especially in the big domain, which is the domain eukarya. So within the domain eukarya, we have the kingdom of animals, and we have the kingdom of plants, but more interesting for us microbiologists are the kingdom protist, which does have only microorganisms. I don't know what happened there. Let me see if I can fix it. Yeah. And the kingdom fungi. So protists and fungi have microorganisms. And then the other concept is that of eukaryotic cells versus prokaryotic cells. All of the living things in the kingdom eukarya have eukaryotic cells, which are the cells that have a nucleus, a membrane-bound nucleus, and membrane-bound organelles. You can see here the membrane-bound nucleus. I'm trying to make my pen work, and for some reason it's not working. Okay, try now. One more time. All right, I don't know why it's not working. So membrane-bound organelles and a membrane-bound nucleus, as you can see here. Okay. There's the nucleus, organelles like mitochondria, Golgi apparatus, endoplasmic reticulum. This right here will be the Golgi. Actually, this will be the endoplasmic reticulum, etc. Versus a prokaryotic cell, which are found in the domain bacteria. Prokaryotic cells are simple. They essentially are a little sac of lipid membrane surrounding a cytoplasm, and everything is found in that cytoplasm like a soup. There are no compartments. The DNA is in the cytoplasm. There are no membrane-bound organelles. There are no inner membranes. Sometimes it's described as a cell that has no inner membranes. It only has the outer membrane. So the domain archaea also has prokaryotic cells. So definitely know the difference between prokaryotic and eukaryotic cells. And know the microorganisms found in the domain eukarya and the domain, which are the protists and the fungi. And then this is just a little aside. In medical microbiology, we do encompass the parasitic worms, which are actually animals. So they're not technically microorganisms, but medical microbiology does study parasitic worms. However, definitely part of microbiology, all the members of the domain archaea, all the members of the domain bacteria. So a lot of PowerPoints explaining to you the different domains, prokaryotic versus eukaryotic cells. This tells you this is important. So definitely understand those concepts. Now, having dwell on the differences, we should also point out the similarities. So what is similar among all living things? This is pointing out the differences, and we should be able to find similarities here in a minute, hopefully. Oh, never mind. Let me go back. All right, similarities. Let's go for that. Very simple. Things that all cells have, regardless of whether they're prokaryotic or eukaryotic. So similarities. Cell membrane. Everybody has a cell membrane. Okay, there may be a slightly difference between eukaryotes and prokaryotes. Thank you, DNA, good one. Absolutely, DNA, definitely. Everybody has DNA. Ribosomes. Everybody has ribosomes. I think that's it. Those are the similarities among all cells. So you could say that is the minimum you need to create a living cell. It's a cell membrane, ribosomes, DNA. Okay. And this picture shows you the difference in size between eukaryotic cells and prokaryotic cells. The huge pink blob, which is kind of hard to see because it's so big, with a nucleus in the middle, that's a eukaryotic cell. It's a cheek cell. Whereas the little purple dots that you see all over are the bacteria. So that gives you an idea of size difference between eukaryotes and prokaryotes. All right. Now, the next PowerPoints are going to focus on each different type of microorganism and give you their characteristics. So we start with the bacteria, the members of the domain bacteria, which are all prokaryotic cells. They're all single cells. And part of the reason why is because we believe a prokaryotic cell is too simple to grow into a multicellular organism. Having said that, there may be some bacteria that do grow appendages to them, which are not multicellular, but are definitely coming close to that. The cells have definite shapes, which is important because we're going to use shape to identify the organism. And you're going to see that very quickly that when you look at a slide of a bacteria, the first question you're asked is, what's the shape? Notice that here it says most have cell walls. Not all, but most. Okay, adverbs are very important in microbiology. Now, a cell wall. Everybody has a cell membrane. So every single living cell in this planet has a cell membrane. Outside of the cell membrane, there is another covering, which is a cell wall. And most bacteria are going to have cell walls, but not all. In most cases, but not all, the cell wall is going to be made of peptidoglycan. You may even argue that in all cases, there is some form of peptidoglycan in the cell wall. That would be a true statement. However, in some cases, the peptidoglycan is not the bulk of the cell wall, if that makes any sense. Now, one thing about peptidoglycan, however, this is bacterial only. And when I say bacteria, I mean members of the bacterial domain. That is the only place where you find this chemical called peptidoglycan. That is a bacterial molecule. The importance of that is our immune system knows it. Our immune system knows that if there is peptidoglycan present, there is a bacteria around, because that is a bacteria-only molecule. The bacteria divide by binary fission. The cells essentially split into two. The DNA is found in the cytoplasm in a discrete region called the nucleoid. They lack cell membrane-bound organelles like mitochondria or Golgi apparatus or endoplasmic reticulum or lysosomes. And those that move by means of flagella. And it says many. So many move, not all of them move. So begin to have an idea of what a bacteria would look like. It's just a little membrane surrounding a cytoplasm with ribosomes and the DNA somewhere in there, and a cell wall. Most will have a cell wall. Shapes are important, so they can be round, they can be spirals, they can be rods. Those that move by means of, will do so by means of flagella, and they divide by binary fission. And that's just a rundown of the same thing. Archaea, we don't spend too much time on archaea. They're not medically important, so we don't spend too much time on them. However, they're very similar to prokaryotes. I'm sorry, to a bacteria. So the domains archaea and bacteria both have prokaryotic cells. That's definitely one similarity. When they move by means of flagella. They divide by binary fission. The differences are going to be chemical. They have cell walls, but the cell wall is not peptidoglycan. Peptidoglycan is exclusive to the domain bacteria. Some inhabit extreme environments, which again tells you they have chemically they're different. Their lipids are linked by different chemical bonds that make them more stable in extreme environments. So they are chemically speaking, they have differences from other cells in the planet. Under a microscope, they're going to look exactly like a bacteria. You will not tell them the difference by just looking at them under the microscope, because again, the differences are chemical. The domain eukarya, we're going to look through the microorganisms in the domain, and again, point out the characteristics. And when you think of characteristics, you're looking at characteristics of that particular group, what is true of every member of that group. So every member of the group protist algae are going to be eukaryotic cells. Every one of them are going to be photosynthetic. Every one of them are going to have a cell wall. Most of the time, the cell wall will be made of cellulose, but not always. So notice that that's not a characteristic, the fact that they have a cellulose cell wall, because sometimes that's not the case. They can be unicellular or multicellular. They are eukaryotes, so they're going to have the nucleus and the organelles. They belong to the kingdom Protista, and they reproduce by sexual and asexual reproduction. Okay. So the reason why we're dividing the protists into algae and protozoans is because medical microbiology is not as concerned about correct taxonomy or correct divisions. They are concerned about identifying something quickly. Algae is not really a medical concern for the most part. Protozoans are. And traditionally speaking, they belong to different kingdoms. It is not but in the past 15 years or so that they have been put under the same kingdom, Protista. Medical microbiology is not as concerned about being accurate as to being effective at identifying organisms. And that's part of the reason why we're still dividing the Protista between the algae and the protozoans. You may not find that division if you were taking a biology class, a true biology, general biology class. Okay, so the protists, protozoans, okay, that's just going over the same. There's pictures of algae. I notice that most of them are green because of the chloroplasts. They're photosynthetic organisms. Okay, more of the same. Okay, protozoans. They're also protists, but they are a bit of a different group, it's at least as far as microbiology is concerned, because some of these can and do cause human disease. So a medical microbiologist should be able to identify them quickly and effectively. Characteristics, they are eukaryotes. That's the first thing. So they are going to have a nucleus, they are going to have membrane-bound organelles. In contrast with algae, which could be unicellular or multicellular, protozoans are unicellular. So that's one difference between algae. There's another difference right here. They lack a cell wall, whereas algae have cell walls. Another difference is that they only reproduce asexually for the most part. Okay, there's a few exceptions, but even those few exceptions obviously will have asexual reproduction. The next one is that they are motile. And they're going to be motile by means of three mechanisms, cilia, flagella, or pseudopods. Some are non-motile, but those who are non-motile have complex lifestyles, and they are motile at one point or another during their complex life cycles. So even the non-motile will be non-motile as adults, but they are motile at some other point in their lives. They're also called protozoans. So they do have a cell membrane, but they don't have a cell wall. They are, in microbiology, oftentimes they're identified by what they look like and what kind of motility they have. So they may have cilia, they may have flagella, or they may have these long extensions of their cytoplasm called pseudopods. And they reproduce asexually or sexually. They're non-photosynthetic. There's another difference between them and algae. Okay. The next one is the kingdom fungi, which are non-photosynthetic. I don't care what color they are. Sometimes they're greenish. That doesn't mean they're photosynthetic. That just means they have green pigments. They're non-photosynthetic. They pretty much get the same type of energy as we do. So they're similar to us in that regard. They're heterotrophs. However, it does say that they degrade substances. Okay, so fine. They spit out digestive enzymes, digest their food outside, and then they eat it up, which we typically don't do that. There are two forms of fungi, mold and yeast. And even though we present those as two definite different forms, in reality, some mold can turn to yeast and some yeast can turn to mold. So there's a lot more plasticity and there's more of a close relationship between those two forms of life than we are led to believe by just telling you molds are multicellular, yeast are unicellular, some are mold, some are yeast. Not quite true. Some yeast can turn to mold, some mold can turn to yeast. Okay. But in general, molds are multicellular, yeast are unicellular. Molds are multicellular. They make filaments called hypha. They have cell walls. All fungi have cell walls. And the cell walls are made of chitin, which is the same substance that insects have in their exoskeleton. They can reproduce sexually and asexually. They make spores in their little, at the end of the hypha, there's typically a fruiting body with spores. So that is reproduction. The filaments, they have a cell wall. This is sexual reproduction. Yeast, on the other hand, are unicellular. They exhibit asexual reproduction in the form of something called budding. Obviously, they have a cell wall. Budding means that they grow a little bud, a little cell right there, which begins to grow and grow and grow until it turns into a big cell and then dislodges from the mother cell. So there's another little bud, there's another little bud. And that's unique to yeast, to the point that when you are in a medical microbiology lab, when you're looking at a specimen of, let's say, a vaginal discharge, and you make a stain of it, a gram stain, and you look through it, you're going to see white cells, white blood cells. And these yeast look a lot like white blood cells. So what you look for is the budding. And when you see little cells budding, then you know, oh, that's not a white blood cell, that's a yeast. Okay, so that's differences between fungi and yeast. They are eukaryotes, so they're going to have the cell membrane, the organelles, the nucleus, etc. And that's a scanning electron micrograph of a yeast. And these little structures here are the places where buds dislodged from the mother cell. Okay. So a lot of this is going to be memorization, knowing the differences and similarities between all these groups. There is a chart under study guides that summarizes all this information. Briefly, we go into the kingdom Animalia. Medical microbiology does look at parasitic worms. Parasitic worms are also called helminths. All worms are helminths. They're multicellular. They're animals. All animals are multicellular. All animals reproduce sexually. They have no cell walls because we animals don't have cell walls. There are two forms of parasitic worms, the flat ones and the rod ones, so platyhelminths and nematodes. Okay, so know the differences between the parasitic worms and other microorganisms. Some of the concepts, the terms you should know. And then this is the chart that I talked about. We have an empty one, and we have the filled version of the chart. All right. Any questions? We only have a few PowerPoints to go, so I'm going to go ahead and wrap it up. Not wrap it up, but finish. Even if we go a little bit past our time, it's fine. I don't have anything else after these. Let me double check, but I'm pretty sure I don't. No, I don't. So I can go ahead and keep going a little bit more. This next PowerPoint looks at nomenclature. How do we name microorganisms? And the lowest level of names would be the genus and the species. So the highest one would be the domain. Then we're going to kingdoms, then into phylums, then families, then class, then order. Did I say phylum? I did say families. I meant phylum, not families. Phylum, KFC, phylum. Anyway, yeah. So at the lowest level, right before family, so we have family, genus, and species. Those are the three lowest levels: family, genus, and species. And so at the lowest level, that we name organisms by their genus and their species. So the genus is the first name; it is capitalized. The species is the second name, which is not capitalized. So for examples of genus, of genera, I should say, would be a Staphylococcus, a Streptococcus, Haemophilus, are example of genera. Okay, so I notice that the names are capitalized. So for example, a bacteria name is Staphylococcus aureus. Staphylococcus is the genus, the species is aureus. The entire name would be either underlined or italicized. We do expect the students to write the names of organisms correctly. So any type of lab report or anything that you write, if you're going to write the name of a microorganism, make sure you write it correctly, or you will be penalized. So again, Staphylococcus, the genus is capitalized, species is not capitalized. Sometimes we abbreviate the name of the genus, especially if you're listing different species of the same genus. You may start with saying, okay, Staphylococcus aureus, Okay, and then the next one will be epidermidis, and instead of writing Staphylococcus aureus, you just write S, epidermidis. And we're to assume that we're still in the same genus. And then you can say, I don't know, Saprophyticus, and then you're assuming that this is the same genus. In the case of E. coli, there is only one species in the genus E. coli. And that's why E. coli, oftentimes we just put the E and then coli, is because there is no other species. So that's, and also because that's probably a very famous bacteria, doesn't need us to write the entire genus. All right, so a genus is a big box that encompasses bacteria cells that have common characteristics. So for example, in the case of the genus Staphylococcus, all of the members, no exceptions, of the genus Staphylococcus are round bacteria that have a cell wall that we classified as gram-positives. They grow in clusters. This would be the arrangement. And they grow with or without oxygen. So they're what we call facultative anaerobes. So those are the characteristics of all of the members of the genus Staphylococcus. There's probably others. These are the first ones that come to mind when you think of a Staphylococcus. So every single species of a Staphylococcus will have those characteristics. One thing to note, because all Staphylococcus are arranged in clusters, oftentimes the word Staphylococcus is used to describe cluster arrangement. I really don't like doing that because to me it's confusing. What are you referring to? Are you referring to the genus or the arrangement? But I noticed that the virtual labs do that. That they instead of saying cluster arrangement, they say Staphylococcus arrangement. Okay, so be aware of that. If they say Staphylococcus arrangement, they're not talking about the genus, they're talking about the arrangement. All right. Species will give us differences between the members of a genus. So, for example, Staphylococcus species aureus, Staphylococcus species epidermidis. Both of them are gram-positive cocci arranged in clusters. Both of them can live with or without oxygen. But when you grow them in a plate, you can tell that one of them has a yellowish color to it, the colonies, the growth. The other one has a whitish growth to them. So that alone tells you, oh, these are definitely not the same as Staphylococcus. Furthermore, when you start doing chemical tests, turns out Staphylococcus aureus can ferment a sugar called mannitol. Yeah, okay, I'll go back to the cluster arrangement. Give me just a second. Let me finish this PowerPoint and then I'm going to come back. Good question. The question is, what is a cluster arrangement? So I'll show you here in just a second. Other differences between these two species, Staph aureus ferments mannitol sugar, lactose sugar, and makes an enzyme called coagulase. Whereas Staphylococcus epidermidis does not ferment mannitol, does not ferment lactose, and cannot make coagulase. Okay. So there are chemical differences between them that allows us to further classify species. Cluster arrangement, what that means is that the cells, okay, this is what it means. It refers to the planes of division. So when a cell divides, it could be able to divide in any plane, in which case the daughter cells will end up being, they kind of remain attached to each other for a while, and they are just in any plane. Some cells, and you end up these clusters that you see here. Okay. Some bacterial cells can only divide in one plane. Let's say right there. That's the only place where they're going to divide, which means that when they divide, they're going to end up making chains. Others may have, I don't know, maybe 3 planes of division. Let's say 2, and then they end up making, so there are, okay, this is not working out for me. So other times they have 2 planes of division, 1 and 2, and then they're making little packages of 4 called tetrads. So the arrangement, a couple of things about arrangement. The arrangement can be very important towards identifying organisms. Sometimes it can be important, many times it's not, but sometimes it can be important. So it happens that in the gram-positive cocci, in the circular bacteria, arrangement is important. That's why it's noted when classifying these groups into genuses. So the Staphylococcus genus have multiple planes of division, so they form these clusters when they divide. The entire population kind of clusters together. Versus a Streptococcus, which only has one plane of division, and when they divide, they end up making these little chains. Okay. Or the Micrococcus, which have two planes of divisions, and they end up making these little packages of 4. So the arrangement is how does the population end up organizing visually as they're dividing, and that goes back to the planes of division. And sometimes it's very obvious that there is some form of arrangement. Most of the time it isn't. Most of the time you may see clusters, singles, most of the time it's not that obvious. But when it comes to the cocci, to the circular ones, it is important. Does that help? Okay, good deal. So nomenclature, we name bacteria by genus and species. Now, here's the deal though. In theory, not in theory, but in reality, bacteria divide asexually. So if you have one cell that makes an exact copy of the exact copy of itself, now you have 2 cells that are exactly alike, and when they divide, now we have 4 cells that are exactly alike, and so on and so forth. Essentially, bacteria clone themselves. So there is, or there shouldn't be, variety. All E. coli in the world should be exactly alike, in theory. We know that that is not the truth. We know that there are variations. So the next question is, how can that be? How can there be variations? Well, there are mechanisms through which these clones may acquire variety, may acquire different versions of their genes. One of those, the most obvious one, is through mutations. There are others that we're going to explore in chapter 8, but mutation is the most obvious one. So we do know now that bacteria can acquire diversity, and then they create what we call strains. So we cannot say that all genus and species of bacteria are the same. There could be slight differences. I'll give you an example. E. coli, Escherichia coli. There is a strain of E. coli which has been named O157H7, which is able to produce a powerful toxin. It has acquired a gene from other bacteria called Shigella dysentery, acquired a gene to make a really bad toxin. So now we have a strain of E. coli that is considered pathogenic and is different. It may be 99% similar to all of the E. coli except for that little difference of that gene. So that is the strain. The problem with the strains is that there are no rules on how they're named. Whoever identifies the bacteria strain gets to name it. So there are all kinds of names for bacterial strains. So a strain is a variation of a species. That's probably one of the best ways to define what a strain is. So it's a bacteria that has the same genus and species. However, there may be 1 or 2 genetic differences, which makes it important to humans. Keep in mind, there may be strains out there that we don't know they exist because they're not important to humans. It's when they become important to humans that they're identified. Okay, so the humans have strains. Yes, of course we have strains. As a matter of fact, each one of us is a strain. The strains are insignificant, are not important in an organism that doesn't clone. We don't clone ourselves. We reproduce by sexual reproduction, which means every single offspring is going to be different, to the point that even identical twins, which you could argue are belong to the same strain, wait 50 years, and those identical twins would have changed their genetics due to variations picked up from the environment. So going to that extreme, there is no such thing as 2 humans that are identical to each other. All right, just to point out go back to this strain, other examples of strains. Haemophilus influenza, it is a common bacteria. There's a T here. A common bacteria in our throats. It's a common, it's a normal flora of the throat. Okay. Despite of its name, it does not cause influenza. There is a strain of Haemophilus influenza called type B. Again, notice that the names of strains can be anything. This one is called type B. And type B, Haemophilus influenza type B, causes meningitis in young children. So there's a strain of a perfectly safe bacteria, which is not safe, it's pathogenic. So, have an idea of what strains are? Last thing we talk about are the infectious agents, which are not cellular organisms, or actually I probably shouldn't even call them organisms. They are what? Agents, molecules that are infectious. There's three types: viruses, viroids, and prions. Probably viruses are the most important as far as human health is concerned. They're called infectious agents because they're not cells. They're not technically alive. They are molecules that are capable of infecting living cells and triggering those living cells into making more of these infectious molecules. The more complex ones are viruses. All of them are obligate intercellular parasites because none of them are cells. So in order to make more of themselves, they need a living cell. In the case of viruses, they can infect any living cell. Well, there are viruses that infect bacteria, viruses that infect plants, viruses that infect animals. They're made of a genetic material, a nucleic acid, whether it's DNA or RNA, and then a protein. So there's two types of molecules in a virus, a nucleic acid and a protein. Viroids are parasites of plants, as far as we know today, and they're just an RNA molecule, no protein, no DNA, just RNA. Prions are parasites of animals, and they are just a protein, no nucleic acids. So, know the difference between those three? And so viruses are acellular, they're very small, smaller than most bacteria by a factor of 100. They have the nucleic acid, DNA, or RNA, never both together, have the protein code. Sometimes they may have a lipid envelope, which is actually the cell membrane of the cellular host they invaded last. So it's not a viral envelope, it's a cell membrane of a cell that they stole. Obligate parasites multiply by using the host. This is a picture of a virus that infects bacteria. Human viruses do not look this cool. Little spaceship, and that's a little bacteria being infected by the viruses. And then viroids are just RNA molecules, and they typically infect plants. Whereas prions are just a protein molecule that typically infects animals. Okay. That is basically it. When it comes to size, I think the best way to do this is to look at eukaryotic cells versus bacteria. And bacteria are in the range of, bacterial cells, I should say, are in the range of between 6 and 10 micrometers, whereas the most eukaryotic cells will be in the range of 20 to 100 micrometers. Organelles, notice that organelles like a nucleus or even a mitochondria are about the size of a bacterial cell. Viruses, the biggest ones, are close in size to the smallest of bacterial cells. Most of them are about 100 times smaller, however. And then you have the molecules, which are obviously much smaller than all of these. Okay. That is it for this chapter. Any questions? The last just goes back and revisits everything. It's a review. All right, any questions? Okay, guys, so we can tackle the next chapter next week. We'll have the meeting for lab on Thursday, and we'll cover lab report 2, which is microscopy. And again, yeah, go ahead.
Professor, I emailed you today about the ebook. I still
Yeah.
can't access it. When I click on it's still not working.
Okay, so you click on the link that I made and it doesn't work for you? And my guess is you have you signed up to McGraw-Hill? Have you done that? Have you signed up? Okay, if
No.
you haven't signed up to McGraw-Hill, then you're not going to be able to access anything. You need to sign up to McGraw-Hill. Okay, so how do you sign up for McGraw-Hill? Let me go back. Let me go back to the. You know what? I'm going to stop